Let's spend a few minutes now and take a deeper dive into this molecule called DNA. Remember, DNA is an abbreviation for something. It stands for deoxyribonucleic acid. DNA is one of two types of nucleic acid that you find in living cells: DNA and RNA. DNA is composed of a building block called deoxyribonucleotides. Now, those subunits, those building blocks of DNA, are sometimes just referred to as nucleotides. What exactly is a nucleotide? Well, it's a combination of three things. It's a simple sugar, it's a phosphate group, and it's a base, or what's sometimes called a nitrogenous base. Take a look at the picture down at the bottom of the slide. What you see in this orangey red color, this is the sugar. This is the carbohydrate portion. In DNA, this sugar is called deoxyribose. So that's where the deoxy part of DNA comes from. It comes from the sugar. You can see that this particular sugar has 5 carbons in it. Now, something to know that's important for DNA structure is that when you see a molecule that has a ring formation like this one, the carbons in that ring are numbered. And it's important for us to know which carbon is which, because there are 2 carbons in this sugar that are going to be important for us in terms of orienting ourselves to the structure of DNA. So, carbon number 1 is the carbon located right at this corner. Carbon number 2 is right here. Carbon number 3 is right here, and you can see it's labeled. It's labeled as 3 with a little mark next to it. We read this number as 3 prime. That's how you read that. Notice that there's a hydroxy functional group attached to the 3 prime carbon. This hydroxy group is reactive. We talked about how functional groups are the groups that come off of chemical structures that are often involved in some part of a chemical reaction. Sometimes bonds are made between functional groups, and the same is true for this one. Now, the 4th carbon in the deoxyribose sugar is right here. And the 5th carbon is actually not part of the ring. The 5th carbon is right here. And you'll notice that that one is also labeled. That one is labeled 5 prime. That particular carbon is also going to be important to us when we orient ourselves in the actual DNA molecule. Now, attached to the 5 prime carbon is the second part of this molecule, which is a phosphate group. Now, they haven't drawn the phosphate group out in its entire structure. Remember, a phosphate group has a phosphorus atom in the middle and then oxygens surrounding it. When we draw a phosphate group, we often draw it as an uppercase P with a little lowercase I next to it. Remember, you saw that when we talked about ATP. We call that inorganic phosphate because it has no carbon in it. The other way to draw phosphate groups is to just put a circle around an uppercase P. Everybody will read that as phosphate group. So, sugar, deoxyribose, phosphate group, and the third part of this nucleotide is the base. Now, you'll hear bases referred to sometimes as nitrogenous bases because they have quite a bit of nitrogen in them. This base, as it's drawn in this picture, is just a generic a generic structure for a base. It could be an A or a T or a G or a C. So each nucleotide, each of the building block subunits of DNA has one of those 4 bases attached to it. Now, on this slide, we can see a drawing of a double-stranded piece of DNA. Now, this happens to be the shortest double helix that you could possibly imagine. It's only got 2 bases in each strand, so it's very, very short. Now, DNA exists as a double-stranded molecule in living cells. Okay? It doesn't exist. It just doesn't show up as a single-stranded molecule. It shows up as 2 strands that have come together and are held together through hydrogen bonding between the bases. This double-stranded sort of ladder structure then twists into what we call a double helix. It has 3 dimensions to it. Now, bases in each of the 2 strands of DNA sort of jut off of those strands, so they come together, they interact with each other, and they hydrogen bond. But they hydrogen bond in a complementary pattern. In other words, it's not that A will hydrogen bond with A. A is going to hydrogen bond with T, and G is going to hydrogen bond with C. There's complementary bonding that goes on between those bases. Now let's take a look at the drawing. You'll notice that you don't see nucleotides in this drawing. I don't see anywhere that there's a sugar, a phosphate group, and a base. I can see a base right here. This green structure is labeled guanine. I can see a base right here. This one's labeled adenine. On the other strand, I can see a base labeled cytosine, and I can see a base labeled thymine. The other thing I can see is that on this strand, the guanine is hydrogen bonding with the cytosine on this strand. I can see that. I can see base pairing going on. The little dotted lines, remember, are how we draw hydrogen bonds. And in this case, they are highlighted in yellow. So I've got a hydrogen bond and a hydrogen bond and a third hydrogen bond holding the guanine and the cytosine together. I've also got 2 hydrogen bonds holding the adenine and the thymine together. But where is the deoxyribose sugar? Where are the phosphate groups? They're not drawn in this picture. And that's because every single nucleotide has a deoxyribose sugar and a phosphate group in it. Every single subunit in the molecule of DNA has those 2 things in common. So oftentimes when we're drawing DNA, we just leave those structures out, and instead we draw what we call the sugar phosphate backbone. So in other words, we have to imagine for this guanine, for this base, there is a sugar right here. There's a deoxyribose sugar right here, and there's a phosphate sticking off right here. Down here where the adenine is, there's a sugar right here, and there's a phosphate group right here. We just haven't drawn them. We've drawn this big, thick red arrow instead. And this represents the sugar phosphate groups, the sugar phosphate backbone of DNA. The only thing that differs from nucleotide to nucleotide to nucleotide is the base. So that's what we draw. And of course, you won't always see bases drawn as molecular structures like this, chemical structures. Most of the time, you'll just see the letter A or the letter G. or the letter C, or the letter T. Now, something else that's important to notice in this drawing. Notice that the sugar phosphate backbones are oriented in different directions, opposite directions. This strand of DNA is drawn so that the head of the arrow here is pointing down, and this strand is drawn so that the head of the arrow is pointed up. First of all, what does the head of the arrow represent? It represents where the 3' hydroxy group would be located. Remember, the 3' carbon is part of the ribose, part of the sugar, and it has a hydroxy group sticking off of it. So when they draw the arrowhead here, they're telling us that the 3' hydroxy group would be located right here. At the other end of the sugar phosphate backbone would be the 5' carbon. This one right here. In other words, this strand of DNA has directionality associated with it. What that means is one end looks different from the other end. One end of a nucleotide looks different from the other end of a nucleotide. So on any strand of DNA, on one end, there's going to be a 3' carbon exposed. There's going to be a hydroxy group exposed or jutting off. And on the other end of that strand, there's going to be a 5' carbon exposed. When you put two strands of DNA together, the direction of that strand is going to be opposite one to the other. In other words, if the 3' end is over here on this strand, it's going to be at the opposite end on the second strand. We give this sort of orientation a name. We say that these two strands are anti-parallel. They're parallel to each other, but they are anti-parallel because they're flipped in their orientation. That's an important thing to know about the structure of DNA. So, the cell is using its DNA for two very different purposes. The cell is going to use its DNA sometimes through replication, just to make a copy of it. And it only does that when it's getting ready to divide. Remember, when cell division occurs, when a bacterial cell or archaeal cell or a eukaryotic cell gets ready to divide, it has to copy its DNA so that each of the daughter cells gets her own copy. The entire DNA molecule has to be copied during this replication process. Each of the two strands of DNA is going to be a template. In that case, the double-stranded DNA molecule will split into two strands, and then each of the strands will be used to make a second strand, a complementary strand, based on that complementary base pairing. A goes with T, G goes with C. Each of the original strands, in other words, serve as a template during this DNA replication process. We give this a name too. We call this semi-conservative replication. That term means that each original strand is going to be used to build a new strand. Let's take a look at this in the form of a drawing. Over here we've got, again, a very short piece of DNA, right? Two strands, and they're twisted around each other in that double helix formation. Now these little sort of sawtooth structures that are coming off of each strand, those represent the bases. And that red line represents the sugar phosphate backbone of each strand. And remember, those two strands will be oriented anti-parallel. So the 3' end on this strand is right here. And if I follow it over, the 5' end is right here. The opposite is true on the other strand. The 3' end is right here. And if I follow it over, the 5' end is right here. The A's are hydrogen bonded to T's, and the G's to C's. Now, this DNA, let's imagine, is inside a cell that's getting ready to divide. And the cell needs to replicate this molecule. The first thing that has to happen is those two strands, we'll call them the old strands, they have to be separated. And that's what we're seeing over here. Here's the top strand. Here's the bottom strand. And notice what's happening. Each one of those strands is being used as a template to build a brand new strand. The brand new strand can be made because the cell can just match up bases, complementary bases. If it sees an A in the old strand, it knows to put a T with it. If it sees a G in the old strand, it knows to put a C with it. So it's going to build one new strand on each of the two old strands. Semiconservative replication. The diagram continues on this slide. We can see the new strands being made again on the old strands. And once the new strands are completed, we now have two DNA molecules. We started with one. We split it apart into two strands, and we built new DNA on each one of those old strands. So I have a double helix here, and I have a second double helix here. Each one of these is made out of one old strand and one new strand.